When you look at plants, the differences seem pretty obvious, right? I mean, a huge oak tree versus a field of wheat. They couldn't be more different. But what if I told you the real story, the most fascinating part, isn't what you see on the outside? It's all hidden deep inside, in their anatomy.
So that's the big question we're diving into today. We're going to peel back the layers and look at the internal blueprint that makes these two giant groups of plants tick. All right, let's get into it.
Okay, so to figure this all out, we've got to start with the basics. We need to understand the building blocks. We're basically going to become detectives and explore the internal structure of plants. The field of plant anatomy. That's where the first clues are.
And it all comes down to this. You have cells, right? And these cells, they team up to form tissues. And those tissues come together to build the organs we all know and love. Roots holding them down, stems reaching up, and leaves catching all that sun. Getting this simple hierarchy is absolutely key to cracking this case.
So if we're going to compare our two main suspects here, monocots and dicots, we need the right set of tools. And for a plant detective, that toolkit is made up of 3 core tissue systems. And the cool thing is, every single flowering plant hasn't. And honestly, it's pretty simple when you break it down. Think of it like this. First, you've got the epidermal system. That's just the plant's skin, its protective outer layer. Then there's the ground system. That's kind of like the scaffolding and all the filler material that does the heavy lifting, like photosynthesis. And the vascular system, well, that's the plumbing. It's a whole network of pipes moving water and food all over the place.
Let's look a little closer. The epidermis. It's basically this single layer of cells that acts like a shield. On the leaves and stems, it's got this waxy coating called a cuticle to keep it from drying out. And down in the roots, you've got these tiny little root hairs that are all about grabbing as much water and nutrients from the soil as they possibly can.
And peppered all over this skin, especially on the leaves, you'll find these incredible little things called stomata. Each one is a tiny pore with 2 guard cells on either side. You can think of them like little mouths that the plant can open and close. It's how they breathe, really, taking in carbon dioxide and letting out oxygen.
Okay, so if you take away the skin and you take out the plumbing, pretty much everything that's left over, that's the ground system. This stuff is the real workhorse of the plant. In the leaves, we call it mesophyll, and it is just jam-packed with chloroplasts doing all that photosynthesis. In other places, like the stem, it's all about providing support and storing food.
And last but not least, we have the plumbing, the vascular system. This is the superhighway inside the plant. It's made of 2 main parts. You've got xylem, which are tubes that pull water and minerals up from the roots, and then you have phloem, which sends all the sugary food made in the leaves down to everywhere else. And these pipes, they're bundled together in groups we call, you guessed it, vascular bundles.
Okay, toolkit, check. We're ready. The investigation officially begins now. And we're going to start right from the bottom and work our way up by looking at the roots. This is where we find our first big clues.
So let's look at a classic dicot root, maybe from something like a sunflower. If you slice it open, you'll see the vascular tissue makes this solid core right in the center. The xylem, the water pipes, often makes this really cool X shape, and the phloem is just tucked right in between the arms of that X. And check out the very center, the pith. It's super small, sometimes it's not even there at all. But now, let's compare that to a monocot root, like from a corn plant. Wow, the difference is night and day. Right away, you see this big, well-developed pith in the middle. And the vascular bundles, there are a ton of them, way more than 6. They're arranged in a neat little ring around that central pith. See the difference? It's huge.
So here's the big takeaway, right? Dicots, they've got that dense core, just a few bundles, and a teeny tiny pith. Monocots, on the other hand, have that big central pith with a whole ring of vascular bundles surrounding it. Just this one difference, right at the base of the plant, shows you how different their internal blueprints really are.
All right, let's move on up the plant and check out the stem. And let me tell you, the difference here is not subtle at all. In fact, it's probably one of the most famous ways to tell these two apart. In a dicot stem, those vascular bundles are arranged in this beautiful, orderly ring. It's so neat. And this organization is their secret weapon. It allows for something called secondary growth, which is just a fancy way of saying they can get wider every year. That's literally how trees make those rings you see when they're cut down.
But in a monocot stem, oh man, it's a whole different ballgame. The vascular bundles are just scattered. They're all over the place in the ground tissue, kind of like sprinkles in a cupcake. There's no ring, no organization, and because they're scattered like this, monocots, like grass or palm trees, they can't get thicker every year the way a real tree can. So the main point here is super simple and really easy to remember. Dicot stems, you've got a ring. Monocot stems, the bundles are scattered. Seriously, if you're ever trying to figure out what kind of plant you're looking at, this is a total giveaway. It's one of the easiest clues you can possibly find.
Okay, time for our last piece of evidence. Let's head to the leaves. These are the little solar-powered factories of the plant world. And as you might have guessed by now, their internal assembly lines are put together very differently.
A dicot leaf is a masterpiece of design, really. Inside, that mesophyll tissue is divided into two special layers. Right under the top skin, you have the palisade layer. These are long, column-like cells just packed to the gills with chloroplasts to soak up as much sun as possible. And underneath that, you get the spongy layer, which is full of air pockets to let gases move around easily.
The monocot leaf, on the other hand, is way more uniform. The mesophyll inside isn't separated into different layers. It's pretty much all the same stuff. And since monocot leaves, like blades of grass, tend to grow straight up, they usually have those little stomata, those breathing pores, on both the top and the bottom surface to get as much air as possible.
And there you have it. We've looked at the roots, the stems, and the leaves. We've collected all the evidence. I think it's pretty safe to say, case closed. Let's just do a quick recap of what we found. This right here, this is the whole secret blueprint laid out for us to see. The differences are just crystal clear. In the roots, you have the solid core versus that big ring. In the stems, it's the organized ring versus the scattered mess. And in the leaves, a super specialized factory versus a more general-purpose one. At the end of the day, these tiny internal differences control the plant's entire life, including whether it can even get bigger over time.
So everything from how a massive tree stays standing to how a simple blade of grass bends in the wind, it all comes back to this internal architecture. It literally defines how they live, and that kind of leaves us with a really cool final thought, right? Now that we understand this blueprint, what could we do if we could actually change it? I mean, imagine engineering stronger crops that can survive a drought, or tougher forests. The possibilities are just incredible to think about. Thanks for being a plant detective with me today.